Today's topic is like we'll be covering so many areas. We will be discussing neuromuscular junction. We'll be watching that what really happens at the synapse. We'll also be talking about certain nerves, right? How they are arranged, and we'll be talking about some poisons. Right? So it will be a good combination of almost everything whatsoever we have done at this point of time. So just take 1 minute. That revision will come automatically. Because as we'll proceed further and further, the previous lectures will automatically be revised, and we'll understand that thing today, that the basic fundamental things that will remain the same. So at sharp 10:30, we'll start with our today's topic. And yeah, I received some mails in which say that we start exactly at 10:30. Yes, we start exactly at 10:30. But yes, in the benefit of many of the viewers, those who are joining late, what we shall be doing is the initial 5 minutes will be a bit slow, and then we'll pick up. Okay. All right. So it's time. Let's start. So that's the topic for today. That is the neuromuscular junction. Describing it in very simple words, that when we say that these are the nerves, right? These are the nerves, and then there would be a muscle. At no point of time there is a physical contact. There is always a synapse which is in between. And there is always a synapse. That means there is a gap. And in this gap, there would be release of neurotransmitters. There will be release of neurotransmitters which will be acting on that particular muscle. So when we are talking about, say, these muscles, so this is the neuromuscular junction. That is the junction of nerve and the muscle. Right? Till this point, what we learned was that from one nerve to another nerve to third nerve, things were going. Even there also there was synapse. Here also there is like presence of synapse. But the difference would be something very typical. In fact, it is a very subtle difference. Say one nerve, that synapse, and we were finding the receptors over here. Here also there will be receptors. Okay. So when we say synapse, so we were having presynaptic neuron, and there was postsynaptic neuron. But over here, instead of that, there would be muscle. Just for the purpose of knowing, Sherrington, he is considered as father of synaptic transmission, because he was the person who said that these are the type of vacuums, from these vacuums neurotransmitters are released. That is what we shall be discussing today at length. So synaptic transmission, and he is also called as the father of neurophysiology. He is also called as the father of neurophysiology. So his work and his contribution in this field is massive. So when we said that neuron synapse, so over here, the difference between, that is when we were having the neuron and the other neuron, and this one is the synapse, right? And over here, this is the neuron, and this one is a muscle. So here also there is a synapse. We can say that this synapse is a bit modified. This is a modified synapse. That's the reason that today we'll be discussing some of the clinical conditions. Now, when we say about these things, we already know that there is a parasympathetic system and the sympathetic system. And for the sympathetic and parasympathetic system, there are specific neurotransmitters. So the entire game, as such, it starts from the brain, from the brain, and from brain there comes the spinal cord, correct? And in the entire spinal cord, from the spinal cord, there are the spinal nerves which are coming out. Spinal nerves. So in anatomy, so this is one vertebra, this one is the second vertebra, these are the transverse processes, correct? And in between there is intervertebral disc, and these vertebrae, they articulate with each other, and over there the nerves which will be coming out, and they are called as the spinal nerves. There are total 31 pairs of the spinal nerves. So starting from the cervical, then comes the thoracic, then the lumbar, then the sacral, and the last one, the tailbone or the coccygeal. The cervical, they are 8, thoracic, 12 ribs, right? So similarly, 12 vertebrae, so these are the 12 spinal nerves. Then lumbar 5, sacral 5, and last one is 1. So 20, 30, and 1. So that is the 31 pairs. What we need to know about this is that whenever we'll be discussing, that one question comes, that so many systems are there. Synaptic, that's right. That's it. So whenever we talk about that, okay, there is parasympathetic nerve and there is a parasympathetic system and sympathetic system, etc., right? And then we'll be discussing in detail about, say, not today, but today we'll just have an overview of it. Then we'll be having extra session on it where we'll be discussing every part of it. But say the one nerve which is coming out, so that nerve is supplying the muscles. It's supplying the muscles. It is supplying the viscera also. It is supplying even some other organs also, other glands also. Yes, actually it happens like that. So in order to avoid that confusion, we'll be looking at the layout. That is how the things are arranged. How the nerve, which is looking like a normal nerve, but it is carrying so many functions. It is going to. The simplest principle, which I always explain it like, that in your house, there would be a pipe. The thing what is called as the conduit. Conduit means this is a pipe, and in this pipe, there would be several wires going on. So over here, there would be switches. Right? On one side, there would be switches, and on other side, they would be connected to either the lamp, or they might be connected to the fan, right? Or it might be some power plug, right, where you connect a device. So the switch one, if it is for light, that means right from the beginning over here, all the way till it reaches to the light, right, there is no one in between. It is all the way one wire which goes and it connects to that pinpoint. Exactly the same way this is for the fan, and exactly this is for the power plug. That means even though looks wise, it looks like that there is one pipe which is going. This pipe is nothing but these are the nerves. And inside these are the nerve fibers. And these are the nerve fibers. So it might happen, and it happens. See, just in a minute, it happens that that particular nerve will be carrying multiple type of fibers. And because of that concept, the nerves have been divided into that, whether it is a sensory nerve, whether it is a motor nerve, or whether it is a mixed nerve. And on top of it, sensory, motor, and mixed. On top of it, we are dealing with the sympathetic and parasympathetic also. So everything, because in from the vertebra, right, we'll find that yes, this is the vertebra, right? We'll find that these are the nerves which are going. Now, where is sympathetic, where is parasympathetic, right? They are just going like this, right? So where are they? Where are they really supplying? Are they supplying upper limb? In order to see this thing, we just take a cross section. Right? Just take a cross section of the spine. Right? So if we take a cross section, it would be something like this. Right? This is the spinal canal. That's anterior. This one is posterior, which we call, say, ventral and dorsal. In other words, right, these are the other names, but otherwise it is anterior and posterior. Simple. Nerves which are entering from, and these are called as the horns. Right? These are called as the horns. They are projected. Right? So we have got the anterior horn, and the posterior horn. You can also call them the dorsal horn, right? Dorsal horn and the ventral horn. It can be said. But there is one extra horn which I use separate color, which is over here. This is called as the lateral horn. Lateral horn. A very typical principle. Nerves, they can enter, whatever the nerve is. Nerves enter through the posterior horn. Nerves exit through the anterior. Period. Khatam baat. This is in, this is out. Now, whatever the games we play about the skeletal muscle, about the sympathetic and parasympathetic and ganglia, whatever we talk about, this is the basic principle. The whole spinal cord and these nerves, they are coming. Then they play their roles, right? From one nerve, few fibers will be going. It would happen. So things which are going out, they will be called as efferent. Efferent. Efferent means exit. E for exit. And in which are coming, they are called as the afferent. We used to remember like afferent means aati hai, right? Efferent means exit. So that way never happened. What our spinal cord is receiving? Our spinal cord is receiving. So in spinal cord, it receives. So it receives. So this thing is called as sensory. Sensory. And this one would be the motor. Okay. To add something to this, again we draw this. And this time, we are drawing slightly more detail. We are drawing it like this. See, this is the nerve. So this is the spinal nerve. One nerve which is going, but it is this, when it exits, then it divides, right? Then it divides. But this is carrying, here it is now. Let's start with this sensory part. So it goes over here. This is what is called as the dorsal root ganglia. Dorsal, because this is dorsal. Right? This is dorsal root ganglia. You can see this is like a relay center. It's called the relay center. These are coming from afar, from quite far. So it should not, signal should not die over here. This is like a relay center, and into the spinal cord. From spinal cord, they start traveling up because it has to go up and it has to inform the specific part of the brain, right? Where, say, for example, this is coming from this finger, right? So that particular fiber. So it is coming from hand. So for that finger, there is a specific representation into the cerebrum, and at that point that yes, there is a pain sensation, right? That pin is getting hit on the first finger. So that's how all the fibers, the way we discussed about the conduit, it happens. Brain response. So signals, they come from the top, right? And they go out. So that means they will be motor. They will go all the way out and they will be going into that nerve and they will be reaching to that muscle, those specific muscle, and it will be acting on muscles. Okay, withdraw, right? Take your hand off. It means that today what we'll be discussing is that we'll be talking about the motor. These are the motor nerves because sensories, when they are coming from all those receptors. So there'll be touch receptor, there'll be pain receptor, there'll be thermal receptors. So all those receptors, they are carrying the sensations, and motor fibers, they will be telling and going to the skeletal muscle that yes, you contract. Skeletal muscles understand only one language, whether to contract or not. But that's it. They don't understand anything else. So if the signal comes, they contract. This is the principle behind it. And one more extra thing which we just add at this point, that is this is the lateral horn. So these lateral horns, they will also be sending their fibers. These fibers, they will be going into this ganglion, right? They'll be going into this ganglion. And this ganglion, right, this is what is called as the sympathetic ganglion. Sympathetic ganglion. And this sympathetic ganglion, from here, the fibers, they can even go like this, and thus now, or they can go up, or they can go down. So this is how the sympathetic chain is formed. At this point of time, just understand that this is the sympathetic ganglion. And we'll be discussing these nerve formations, right? Akshat, which diagram you are talking about? Which diagram you are telling? You are written a partial. So, right. So this is how it really goes. So think it like that today we are talking about this motor. Because see, the nerves will be going to viscera also. If there is any pain, right? If you consume too much of food and there are stretch receptors, so in which the stomach lining is stretched, so we feel pain, right? There is pain. So those stretch receptor, that pain will also come and that will be sensed, and accordingly the things will be. Red line is nothing but this is the autonomic system. Autonomic. Right? Sympathetic or parasympathetic systems, they go via this lateral horn. Right? Lateral horn. And for lateral horn, this is like a relay center. This is the sympathetic ganglia. This itself is a big thing, sympathetic ganglia, because some fibers, they will be going up and then they will be exiting from the anterior, anterior path. Some fibers, they will be going down and then they will be exiting, and some will be going directly. So, and then there are say gray rami communicants and white rami communicants, and then there is general visceral afferent, spatial, that general somatic afferent, and similarly efferent. All those things we'll be learning when we'll be learning the formation of nerves. But this is in general that you should really know that that it is the anterior part, which is motor, and the posterior horn, that is our sensory. Okay. The next, now we start with our, before that, as it was asked, This is a simple explanation. Cervical and the sacral, top and the bottom. Right? That is what is called as the cervicosacral outflow. Right? It is called as the cervicosacral outflow. So that outflow is for parasympathetic. It is for parasympathetic. And when we say this one would be what? Thoracic, and this one would be lumbar. So thoracolumbar, right? Thoracolumbar. That outflow, that is for sympathy. That is for sympathy. So it makes picture very clear now that when we talk about the thoracic, that means those nerves which will be coming out, right? And they will be forming that sympathetic ganglia, and then they will be supplying to heart, right? So all of them, cervicosacral, right? I said that cervicosacral. So this is the cervical, this is the sacral, they are parasympathetic. So they are all, the coccygeal, that is just a single nerve. So it is, basically it is cervical and sacral. The next part is now we start with our today's actual session. This was the foundation. See, this is what we have already learned, that this is the resting membrane potential, and in case of resting membrane potential, it is because of the sodium potassium ATPase pump and the potassium channels, leaky channels, right? Leaky channels. This is the resting membrane potential which is generated. Then as the stimulus comes, right? As the stimulus comes, this plus is the stimulus. That means if someone touches, right? If that touch is absolutely light, then there would be few sodium channels, they will be generated, and then signal would die out. Right? Because over here, the signal was not at all strong enough to generate the action potential, right? The higher action signal just went up and it died. Over here, this is what is called as the threshold potential, right? It is called as the threshold potential. So when the signal is strong, right, the enough number of sodium channels, and this is because of the mechanical stimulation, right? They led to sodium influx. And it is at this point when there is voltage gated sodium channels, they fired up, right? And that increased the voltage, right? At this height, the sodium channels, they are deactivated. And then there is activation of voltage gated potassium channels, and the potassium, as it starts leaving, the voltage drops, right? Voltage drops, and it drops even below this RMP, resting membrane potential, and this is what is called as the hyperpolarization. And then this is the time when there is a reset mechanism, which we understood that how the reset mechanism works. Over here also, it is the sodium potassium pump and the potassium pump. They work. In case of say cardiac muscle, there were even the calcium channels. So this is the normal function, right? That's how all the things they really go. The resting membrane potential minus 17. That was the firing point. Right? That was the starting point. And then at minus 60 millivolt, the threshold went all the way up and it's continued. This thing, it is happening. So if we talk about, this is the neuron, right? This is the cell body. This is the cell body. These are the dendrites. Right? These dendrites, they will be forming synapse with everyone else. In between, there were myosin. There will be myelin. Right? There will be myelin, which was forming was increasing the speed. But all we need to know is it is this wave of this action potential, right? That will keep on triggering. It will keep on triggering one after another, and it will reach till the terminals. So this is now, we have got the now terminals. This is the terminal. It is this area. It is this area now we are into. This is the area now we're talking about. So this is nothing but this is the end terminal, or the end point, or the terminal point. So this is the now terminal. Till this point, there is no change. It is just all the way axon, right? Those action potential, they will be reaching to the end. Now over here, we require certain things, and that certain things will change the orientation for our ease. So this is, make it big. This is the terminal end. This is the terminal. Still. Right, so this is the terminal end. Now at terminal end, as the sodium comes, it comes over here. When it reaches at this point, again over here, it is the same sequence, right? The same action potential. Right over here, right? It will go up, then this process continues. But over here, we need to make one more change, and that change is that there has to be the release of neurotransmitters. So first we'll just understand the release of neurotransmitter, then we'll be going to it. So here it is, all the neurotransmitters, they are present in the form of those bags, right? These are called as the vacuums. So all of them, they are into the form of bag, or they are filled. So we can say given the cycles, they are filled. So they are filled with the substance. When they will be empty, we'll see that what happens to them. So right now they are all filled with, say, acetylcholine. They are filled with acetylcholine. We need to release them into this space, because this is the opposite end. There is a space, and this space is synapse. To release them into the synapse, this one is the synapse, we require one, calcium. This calcium is needed. These are filled with neurotransmitter. I wrote acetylcholine, right? So this is the neurotransmitter. Potential, they don't change with age, but you'll get the answer today. You'll get the answer. Just let it go. Only the release of this neurotransmitter is not sufficient. Even the receptors, they are required, so that we'll see that. So here it is the neurotransmitters. What we really need is the calcium. So when the calcium, if it can really enter, then the calcium, it can trigger the release of these neurotransmitters into the synapse. But there is one more thing which is needed, and that is there are the specific spatial proteins are needed. Spatial proteins. Right? These proteins, they are called as the snare proteins. Snare proteins. So unless and until we don't have these two things, it won't work. Right? Simply, it won't work. That means when we are understanding this, we need to understand it in slightly more detail, because the diseases would be associated. Diseases would be associated with it. So if this thing slows down, so then that particular movement, right, that the speed at which you are giving punch, right, it would be slowed down. Because if things are coming slowly, so there will be slow contraction. And When they say reflexes are fast, so reflexes are fast, or so that is simply because of that the whole process is working much faster, right? Sufficient amount of calcium are there, sufficient amount of proteins are there. So when it comes, these vesicles, right? They come over here, then this membrane is absorbed, right? They merge, and then these neurotransmitters will be released. This process is called as the exocytosis. I just write it. We are about to discuss it. Exocytosis. That is the process. Right? That is the process of exocytosis. So for exocytosis, that is exocytosis of obviously neurotransmitters, right? So of, I'm just writing HTML. So understand it is a neurotransmitter. So two things are needed. One, we said the calcium is needed, and second, we said that there are specific proteins which are needed, and they are called as the snare protein. To be very specific, we require a protein, that is the specific protein. It is called as the synaptobrevin. Synaptobrevin. This protein is needed. So let's go back to two concepts, and then we'll see the whole process right from the beginning till end, that is formation of acetylcholine, storage into vesicle. Right? What is needed, man? What's the problem? Everything is getting written. Don't spam. So what I'm telling is that first we understand those principles, right? And then we'll be starting with all the way, right from the beginning, that is the formation of acetylcholine, all the way till it is released, till it is broken, then it is recycled, all the steps they will come out, right? And don't worry, what I'm telling you is that when I'm explaining something, I'll definitely write. I've got the habit of writing. I'll write. But when I'm explaining something, first listen to that. Right? I'll not leave anything unwritten. Today we'll do the revision also, as there were 2-3 emails about the revision. Today we'll do the revision. Right? So if I forget, remind me. We'll do the entire revision at high speed. Now see this. This is one principle. What was this? This was cell, right? And what was this? This was the end, right? Terminal, right? Not drawn proportionately. This is the cell. All the proteins, whatsoever is required, they all are formed here only. So even when we say that some proteins which are needed over here, that means where are they formed? They are formed at the cell. Cell is the factory, right? Cell body. Right? Cell body, this is a factory. So all proteins formed, manufactured here. And then they travel all the way and they reach to this end. They reach to this end. There's only one change. The acetylcholine that is formed here. Right? So acetylcholine, that neurotransmitter, that is formed at this terminal end. Otherwise, all proteins, whatsoever, we'll be discussing several proteins. All those proteins, they'll be formed at the top. They will come over here, they will act. Right? So this is one principle, the first principle. The first principle. And this one is the second, the acetylcholine, that is not synthesized into the cell body. It is synthesized at a terminal. Acetylcholine formed at terminal, and then it is stored. Right? And then it is stored. Then it is ready to release whenever we need it. Yeah. So now we'll make, this is our one end terminal. Right? So from top, the needed proteins will be coming. So let's start with GIT. we consume some food. Right? So from GIT, from that food, choline would be formed. Choline formed. That choline, it will travel through the blood, and thus it will be reaching to every this space, right? Extracellular fluid, right? It is now into this ECM, extracellular fluid has reached every part of it. Now over here we have got one specific protein. Now comes the first protein. This is, I'm giving the number, or let's put this as number. This is the first step. This is the first step. Choline formation, and choline is waiting in the ECM. There comes the protein. This protein is nothing but it is like a gate, right? Because this is the cell membrane, right? This is the cell membrane. Cell membrane of what? Cell membrane of neuron, right? So that neuron is having some integrated protein. This is one of the integrated protein. This integrated protein, its name is so simple because it will be transferring choline. It is called as the choline transporters, right? So that is what is called as the choline transporter. Easy to remember? Transporter. As soon as it is there, so this choline, right, it has enters. So now choline is here. Great, right? So we have got choline. We need to make, remember, acetylcholine, right? Now, the second one in this, if I draw this, then what am I drawing? Which structure am I drawing? That's right. This is powerhouse, mitochondria. Right, mitochondria. That's right. So this mitochondria will be producing acetyl coenzyme. This will be producing acetyl coenzyme A. So we have got one more structure, acetyl coenzyme A, and we have got choline already over here, right? Both of them, they would combine, but as such, they cannot combine directly. They'll need something. So again, there is one enzyme which is needed, and that is called as the choline acetyl, as such, just choline and acetyl, right? And it will be, its name is transferase. Whenever it ends with ase, that means it is an enzyme. So in the presence of choline acetyl transferase, acetylcholine A, coenzyme A, and the choline, it makes our so much demanded acetylcholine. But this acetylcholine, right? This acetylcholine, the moment it is formed, these are the blank bags floating. Immediately, there is another protein. And that's why, remember, all these proteins, they are coming from the top. None of the protein which is formed over here, right? It was just acetylcholine which was formed over here. So this is what is called as the transporter protein. This is once again a transporter protein. So this transporter protein's function is, the moment acetylcholine is formed, immediately pick it up and send it inside the vesicle, right? So this vesicle, it keeps on getting loaded. It keeps on getting loaded. And there would be several such vesicles which will be floating. And this is the beginning. This is where things will be waiting. Right? Things will be waiting, and thus these are our all vesicles, and just consider that all of them, they are now loaded with acetylcholine, ready to launch as soon as the signal comes. So this is the basic preparation. Now, next. Same situation, and we'll draw filled with acetylcholine. These are the vesicles. As the signal comes, same would be the pattern, but it is at 40. It is at in between. Sorry. It is in between. There would be, these are the calcium channels. These are the calcium channels. And these calcium channels are called as the depolarization sensitive calcium channels. So when the depolarization wave would come, right, because from top the signals are coming, that sodium will be coming, right? The wave will come, right? The wave of positive, right, it is coming. It opens these calcium channels. So the calcium starts entering. So this is the calcium, and there will be several such channels. Right? So the load would increase. So the calcium channels, they start coming. So that's why these calcium channels, they are vital, because these are the calcium channels which are triggering this entire mechanism. So this calcium channel has arrived. Now, what has happened to this? That the vesicle which is over here, the vesicle which is over here, this one, it has got calcium sensitive over here. There is one. Once again, there is a protein. And this protein was formed from the top. So this means that this membrane of this vesicle, these bags, they are not bulky, they are very valuable, right? Because they are produced from the top. So that's why we just cannot waste them. So here is the mechanism. This is calcium sensitive. Right? This is a calcium sensitive protein. The calcium sensitive. So this calcium sensitive, in the presence of this calcium sensitive protein, as soon as this calcium comes, so these vesicles, they will start traveling. Right? They will start traveling and they will come near the membrane. Right? They were floating, but in presence of calcium, first thing what really happens is that it comes near the surface. Now, when it comes near the surface, which part? Can you repeat this part? Which part I missed? Anyway, I'll be repeating entirely. So when it comes, when it reaches to this point, there would be now the process of exocytosis should occur. Exocytosis. So we see that what really happens in exocytosis. So two types of proteins are over here. One type of protein is here and one type of protein is here. This is the protein, what is called as the synaptobranine. Synaptobranine. And this one, which is on the surface, it is called as the syntaxine. Both these proteins should hold hands of each other. Both these proteins. So when the calcium sensitive proteins come, it will be triggering the movement of this entire vesicle towards the surface. Now, as it approaches towards the surface, these two proteins, they will join, they will hold a hand, and it will start pulling near the surface. So situation will go something like this. Here it is. It will come, then here it is coming near, then it will be touching it. These proteins, they have anchored this vesicle, and then this membrane that will fuse. So situation will be like this, and then this membrane would shift so that it will be neurotransmitters are released. Acetylcholine released. It means when synaptobravin and syntaxin, when they come near each other, right? When they come, this is evagination, right? The invagination is coming inside. This is evagination, or this is classically called as the exocytosis. Whenever we are talking about the secretions, it is called as the exocytosis. So over here, so the membrane, they fuse and it releases this. So now we have got the new membrane. This entire membrane, this much. If I draw it like this, if I draw it like this, if I draw it like this, right? And if I this is what has really happened. And the neurotransmitters, they are released. And then again, the situation would be like this. Like this. You got it? Because see, this portion, right? This portion was so that was of that vesicle, na? Hai na? So this is see this shaded thing. This is the vesicle. This is the vesicle. So okay. Now it has straightened, but still this portion is the vesicle. We cannot let it waste. Right? We cannot let it waste. So there is one more protein. There is one more protein. So that protein is called as the clathrins. So what clathrin will do? Well, here it is. Here it is. So these clathrins, they are called as the clathrins. They will hold this, and they will hold this. Right? And they will start pulling it. They'll start pulling it. That this is my membrane. They give me back. So the formation would be now like this. And at one point, like this. This is the vesicle. This is the vesicle. And then finally, it will become like this. So we have now the empty vesicle with us. So thanks to protein called clathrins. So function of clathrins is the recovery of vesicles. The recovery of vesicles. So that is also not wasted. So starting from here, it was GIT to choline, which comes into extracellular fluid. From there, it went from there by the choline transporters, it went inside, and this is the choline. Mitochondria produce acetylcholine enzyme A, combine both of them, and that is choline acetyl transferase, and that will be helping to form the acetylcholine. This acetylcholine will be going into the vesicles, right? Because of the presence of this transporter protein, and thus it will be going into the vesicles, and they will be stored over there. As the action potential from top, that is from the axon comes, these are the calcium channels. These are the voltage gated calcium channels which will be activated. They are also called as the depolarization sensitive calcium channels. So when the depolarization wave comes, right, so this depolarization wave, that will be opening all the way these calcium channels. Right? Once these calcium channels are open, so on these vesicles, these are the calcium sensitive proteins. Right? These calcium sensitive proteins, they will start pushing all these vesicles towards the surface. Right? Wait, I'm coming to that. So it will start pushing towards the surface. Now they are having two types of proteins. On the surface of the vesicle, there is synaptobrevin, right? So there would be synaptobrevin on the surface, and on the cell membrane, there is syntaxine, right? There is syntaxine. So you can say that syntaxine and synaptobrevin, they will be holding hands, and then it will be pulling, right? They will be pulling. When it comes over here, process what is called as the exocytosis. So over here, you can say that this is like syntaxine. This is like syntaxine, and this is, you can say, these are like synaptobrevin. It's synaptobrevin. So syntaxine and synaptobrevin, they will join hands, right? And then it will pull, right? When it will pull, these layers, they would fuse, and then this layer will give space, right? It will open up. This place will open up, and our acetylcholine, that would be released. And once again, the surface is reoriented. But we need to take our vesicle space back, right? Vesicle space back. Because some portion of the vesicle is lost. That would be, again, it is a protein that would be formed by the cell, and it will be sent. But why to put extra load on cell membrane? And so we just say whatever is damaged, right, send only that much, rest will recycle. So there comes the clathrines. So these are also proteins. So these clathrines, they will start pulling from both the ends, right? The vesicle membrane, only the vesicle membrane. So this is our vesicle membrane. So it would be pulled and it would be squeezed out, right? It would be pulled and it would be squeezed out. So that's how it really forms. And our new vesicle is ready. New vesicle is formed. And the whole process repeats. So this is how it really goes. One more thing. Now acetylcholine. This is our muscle end plate. This is our muscle. On this muscle, there will be nicotinic receptors. These are the nicotinic receptors. So this acetylcholine, that will be acting on it, and when it acts immediately, it will be sending a wave of action potential inside. When this action potential wave is sent inside, the muscle starts contracting. Acetylcholine is sitting over here, is constantly stimulating. And acetylcholine, there is so much of acetylcholine into the synapse. So if it keeps on moving, it will keep on triggering these nicotinic receptors. So there won't be any relaxation. So no, we need to stop this thing. So there comes one more enzyme. So that enzyme would break the system. Now breaking the acetylcholine, that is fine, but we need to AP is action potential. Yeah. So this acetylcholine is broken. It is broken. It is broken in by cholinesterase. Right? It is broken by cholinesterase. It is broken into acetyl plus choline. Now, out of this acetyl, it is a very sasta product, right? It can be made very easily. So this is diffused out. And this choline, it is taken back. It is taken back. It is recycled. This choline is absorbed back so that it can be utilized once again. You got it? So we can write over here that this is the choline, acetylcholine, and the acetyl. This acetyl diffuses out, this choline is taken back so that it is recycled. So nothing is getting wasted. The next is that when we said that this is how the action potential would start and keep on continuing. Looking at now the clinical aspects of it, all the clinical aspects, then we'll go for the clinical aspect first thing. Those who eat non-veg. So there is a fish called puffer fish. There is a fish called puffer fish. Now in this puffer fish, sometimes, not every time, they say that in the 5-star hotels, those cooks who are expert in making this puffer fish, they are given very high pay scale. Reason that in puffer fish there is one part of the fish, one sting, which is to be removed because it consists of a very powerful toxin, and that is called as the tetrodotoxin. And it is called as the tetrodotoxin. Now this toxin is fatal. This tetrodotoxin, it comes from where? I mean, it comes from this puffer fish, but it acts where? In 1 second you will know. If I say it blocks voltage gated sodium channel, done. Because the moment you say, okay, it blocks voltage gated sodium channels, they are blocked. So that means there is no action potential. If there is no action potential, so there won't be any trigger of calcium. If there is no trigger of any calcium, so there won't be any release of neurotransmitter. If the neurotransmitters are not released, so nothing goes on to the muscle side, and that leads to muscular paralysis. Clear? Do I need to write, or it is self-explained? X means closed, X means blocked, right? So this would be our every time, right? I am writing this blocks so that we understand, but otherwise, as we'll be studying together more, whenever I write like this, that means it is blocked. Where is it present in? Say, if this is the fish, right, there is on its scales, there is a certain area. But no one would ask that. But this is like this tetrodotoxin. It blocks voltage gated sodium channels. So yes, once this is, so then there won't be don't ask much about the power, because I understand it only from the purpose of tetrodotoxin. So this is, yeah, but this puffer fish, say AP is action potential. I'll write it so that there is no action potential, right? If no action potential, so then those calcium channel, voltage gated calcium channels, they won't be released. And if not that, so then acetylcholine, that won't be released. If that is not released, so there would be no muscular contraction. And which muscles? Apart from other muscles, the important muscles will be respiratory system muscles. The respiratory muscle paralysis. And that leads to certain death. So this is one condition. If you can, then it blocks. In humans only we are talking about. But all these conditions whatsoever we'll now be discussing, they're all present in the humans. So this is the first thing, that is the tetrodotoxin. Let's move on to the second condition. Now, this second condition, there is a system in which there is, we'll be learning it in super detail, the immunology. Not respiratory only. Obviously, hand muscles, leg muscles, they will also be involved, but it's okay if one is not moving hand or legs. But what about the respiratory muscles? They will be the affected, they'll be the most dangerously affected. As the respiration stops, death. All the muscles would be affected. All the muscles. But before person gets a chance that his hand is not moving or legs are not moving, respiration has stopped, so it will be the immediate death. When we'll be discussing the immunology, this is that topic which will take about 1 month to discuss, right? It will be discussing it in that much detail because it is this topic which is like most confusing, but once known, you'll be able to understand the hematology, you'll be able to understand all the diseases so precisely. Now, in that, there is antigen. Antigen means pathogen. Pathogen means that bacteria, that virus, right? Bacteria, virus, fungi, right? Virus, fungi, right? All those worms, which are called as the parasites, all of them, right? They are causing some pathology into the human body. So they are called as the antigen. Body fights against it, right? Body say, right? Let's form the antibody. But this antibody formation, it takes 7 to 10 days. Right? 7 to 10 days. And then comes the immune system. Chalo learn. But by that time, there is antibody kills antigen. That's right. Absolutely. And these antibodies, you have to remember, they are called as the, medically, they are called as the immunoglobulins. Globulins. So globulins, immunoglobulins, once again, apna dear protein. Again, a protein. And these proteins, immunoglobulins, remember it like Gamdi. So IgG, so when we say IgG, IgA, right, IgM, IgD, IgE, all the way. Out of these, it is the IgG and IgM, they are very important. They are very important because they will be the first fighters. Achha. Apart from this, so right now this much is sufficient. Apart from this, there will be several support systems which will be coming. Support systems. They are called as the complement protein. Complement system. There is a complement system all the way to C1 to C9. Right? C1 to C9. And then once again, there will be like C3A, C3B, C4A, C4B, C5A, C5B. Right? The point is just one, that this is such a bulletproof system which is keeping our body intact. Otherwise, everyone is getting infection every day at every point, but body keeps on fighting. Now, I'm explaining, say, this small concept, and this concept will suddenly open your eyes. Oh, such things happen. There are few cells which are called as the pluripotent cells. Right? Pluripotent cells. Pluripotent cells means they don't know what they will become. Whether they'll become doctor, they'll become engineer, they don't know. This pluripotent cells means as they will progress, right? They will divide, they will mature, and some of them they will become RBC, red blood cell. Some of them will become WBC, white blood cell. Some of them they'll become platelets. Right? So for the blood formation, these are the pluripotent cells which are sitting into the bone marrow, the red bone marrow. Right? When the kid is born, almost all the bones they are red. And then they become so RBC, WBC, and in WBC again the granulocyte and agranulocyte, and then there are neutrophils, basophils, eosinophils, right? And then there are lymphocytes. In lymphocytes, once again, they divide. Right? There are T cells, B cells, natural killer cells. Natural killer cells are the first line of defense. The moment anything occurs, they immediately come and their work is to kill them. Right? Whosoever the bacteria is, just kill. But when it is finding no, it is becoming difficult, then it make a call. Kya madad ke liye aao. Then T helper cells, they will wake up. Right? And then in T helper cells, there are T helper cell type 1, type 2. Type 1 would say, कि नहीं मैं नहीं आ रहा, मैं मेरे दोस्त को भेज रहा हूँ, right? He is powerful, so he'll also go. T helper cell 2 will call B cell. B cell is like a big boss, tagda gunda, right? He will go and he'll start knocking off. So that's how the whole process occurs. This is such a powerful. Why am I explaining this? Such a beautiful system, but won't this system act against our own cells? Yes, it can. Right? It will act. This is the important thing, that those pluripotent cells, right, very enthusiastically they made RBC, WBC, platelets. But this is our RBC. On the RBC, there are receptors, and on these receptors, these complements, they can come and they can sit, and then they will start acting. This is the RBC. It can really break this RBC. And when this RBC is broken, it is what is called as the hemolysis. Hemolysis, right? Lysis means breaking. Hemo means blood. Blood starts breaking. RBCs, they start breaking. When the RBCs are breaking, right, hemoglobin will come out. Correct? Hemoglobin will come out. That hemoglobin will go where? It will go into the kidneys. Right? Now it goes to the kidneys. See how dangerous things can really be. It will go into the kidneys. Right? When it goes into the kidneys will say, ke chalo, isko fit. Right? So this is how the kidney is. Right? The unit is called as the nephron. So here is the Bowman's capsule. Right? This is what is called as the Bowman's capsule. Proximal convoluted tubule. Loop of Henle. Right? This is the loop of Henle. And then there is distal convoluted tubule, and then it goes into the collecting ducts and further. So when these are the afferent and these are the efferent, and this is the tuft of capillaries, right? So blood comes over here by the process of ultrafiltration. This hemoglobin, it enters, because now the hemoglobin's molecular weight has decreased. It has become chota. So it comes into this proximal convoluted tubule, PCT, right? So in this proximal, convoluted means something which is tortuous. It is convoluted tubule. So when it is over here, this PCT, proximal convoluted tubule, makes a big blunder. It thinks, it is such an important, right? So let me take it back. It tries to take it back. It tries to reabsorb, and it comes into this interstitial space. Because right now it is still in kidney. When it comes, the hemoglobin breaks into heme and the globin component. Globin is protein, but heme is what? Heme is iron. It is iron. And iron in these spaces, it is super reactive. It is super reactive. It destroys tissue. Right? It destroys tissue. So this super reactive iron, it will destroy kidney structure. It will destroy kidney. To be specific, it is what is called as the tubular, because it is into the tubules, tubular necrosis. Necrosis means surgeon. Necrosis, that's destruction. Tubular necrosis, such dangerous things can happen. Right? Such dangerous things. And everything started with simply because on our RBC, there were few receptors, and those receptors, that was a gift by pluripotent cells. And why those receptors are needed? Well, they are needed because when the bleeding occurs, so then all of them, they should come and join together so that they can form the clump, right? They can come over there. Right? Similarly, it is on platelets. Similarly, it is on WBC. Everywhere it is there. So that's the reason there is one wonderful mechanism which has been generated by pluripotent cell. there is one gene, right? It will say that I'm teaching all of you, right? It will say that all of you are my kids. And as you go further, I'm telling that always remember that whenever the compliment comes, I'm giving you all a gift, and that gift is the surface cutter or the complement destroyer. So whenever a complement comes and attaches to you, immediately destroy. So for that, there is a specific type of protein which is given, and that protein is generated by this gene. It is called as the PIGA gene. Right? PIGA gene. So this PIGA gene, it keeps on making this mechanism, and it keeps on teaching to RBC, WBC, platelet, everyone. Right? So everyone understands that when the complement comes, cut it. When the complement comes, cut it so that this RBC is not destroyed, this WBC is not destroyed, this platelet is not destroyed. Right? So nothing happens. This happens every day, every minute, every second. But this PIGA gene, right, it can be mutated. And it is not a congenital, it is acquired. Right? There are specific poisons, there are specific conditions, right, which we learn in immunology. But in those conditions, this PIGA gene is mutated. When it is mutated, so then this PIGA gene becomes Gajini. Right? He forgets that I need to tell them to make this complement destroyer. So next batch of blood when comes, because see, the life of RBC is 120 days, right? One intelligent RBC who has understood this process has come. But now something happened to PIGA gene. If something happened to this PIGA gene, so the next batch which is coming, it is not having any clue about this complement pattern. Correct? Because our blood is replenished every 120 days, full blood is replaced. All RBCs. WBC life, just 2-3 days. Right? If it goes into the tissue, that's 7 days. Platelets, again, short lifespan. So 120 days, this is the maximum. So even if anything happens to PIGA gene, someone's PIGA gene is affected today, till this batch is over of 120 days, nothing would happen. But all the subsequents, right, they will be having a problem and they can cause this disease. Right? When we say acquired means it is not present right from the birth. It can happen because of certain things, and we'll just have a look at it, that what can really happen. So this is the immune system. I explained this thing in slightly details, that what if this thing goes wrong by certain systems. The thing what is called as the autoimmune disorder. Right? So this is autoimmune disorders. On autoimmune disorders, say for example, systemic lupus erythematosus, right, SLE. This is one of the very, very bad condition. Almost everything is affected. Skin is affected, the lungs are affected, brain is affected, muscle affected, kidneys are affected, liver affected, right? Metabolisms, they are haywire. But here we are talking about one autoimmune disorder. And this autoimmune disorder is doing what? Again, I'll just draw and instantly you will be able to figure out that yes, what we are talking about. Here is our end. Right? Here is our the terminal part. In that terminal part, there were voltage-gated calcium channels. What if this particular disease, it destroys these channels? Now what happens? If this is destroyed, this we are talking about presynaptic disorder. Right? We are talking about presynaptic disorder. Because synapse is here. Now. Right? Here is the synapse. So this presynaptic disorder, voltage-gated calcium channels, they are destroyed. If they are destroyed, that means there won't be any calcium going in. That if the calcium is not going in, so over those vesicles, they will keep on moving inside, loaded with acetylcholine, but it would never be released. Will never be released. This is the condition what is called as the Lambert-Eaton syndrome. Lambert-Eaton syndrome, this is a presynaptic disorder. Here is the postsynaptic. This one is the postsynaptic. Over here, you were having that nicotinic receptor, and this was our receptor. This was our receptor, and this was nicotinic receptor. What if there is one disease which comes and it destroys nicotinic receptors? This is gone, right? It develops, and how it happens? It develops antibodies. Because the antibodies are formed against our own cell, it is called as autoantibodies. So now, in this patient, he has got autoantibodies against nicotinic receptors. Over here, Lambert-Eaton was doing what? It was creating autoantibodies against voltage-gated calcium channels. Two, and this is what is called as, you must have heard this name, myasthenia gravis. Myasthenia gravis. So in myasthenia gravis, there is formation of autoantibodies, right? There is formation of autoantibodies against nicotinic receptors. And in case of Lambert-Eaton, there is formation of autoantibodies against voltage-gated calcium channels. In both the cases, final thing is muscle weakness. Final thing is muscle weakness. So in one case, it is presynaptic, and in other case, this is the postsynaptic disorder, right? So what would be the final effect? Well, the final effect would be muscle weakness, which keeps on getting worse, and then goes into the state of paralysis. When the result is same, so then why we need to know that this is presynaptic or postsynaptic? Treatment. In such patient, in the patient of myasthenia gravis, whatever we do, right, even if we give tons of acetylcholine, there won't be any improvement. But in case of Lambert-Eaton syndrome, if we can artificially give acetylcholine, then they will start functioning. If we give acetylcholine, then they will start functioning. So this is the difference in the treatment, that when we know that where it really acts, so it will make a big difference. From this point, let's learn about some poisons. These are some of the most dangerous poisons of the world we are talking about now. First, we'll start with something which is very soft. But this poison, which is called as the organophosphorus poisoning. What happens in Lambert? I wrote it over here. Formation of autoantibodies against the voltage-gated calcium channels. When the voltage-gated calcium channels, when they are blocked, so now you know. Politicians are not given organophosphorus. While politicians, they are given another poison. We'll discuss that. In fact, there are some other now gases, sarin, that is one of the organophosphorus. So you just in a minute will come. So result will be what? Result will be what? Result will be same. Nah? That's what I said. Muscle weakness. Because whether the acetylcholine is not released or acetylcholine is not utilized, in both the cases there won't be any muscle contraction. Okay? Now comes the organophosphorus poisoning. Organophosphorus poisoning, it is bad because this is the poisoning which is also present into most of the pesticides. Right? It is most of the pesticides. So all those pesticides nowadays, knowingly, unknowingly, that's why they said that we need to wash the food very properly. Or sometimes those into the cold storage, right? They put so much of chemicals. All those chemicals, they are basically poisons. What will they really do? This organophosphorus poisoning or this organophosphorus component, it will block choline esterase. It will block choline esterase. So now what will happen? Now think it from the perspective of that when the choline esterase is blocked, choline esterase was doing what? What choline esterase was doing? What was the function of choline esterase? Choline esterase was chopping the acetylcholine. That's right. It was breaking acetylcholine. Right? It was breaking acetylcholine into the acetyl and the choline. But when this is not happening, so then what? Right? When this is not happening, that means this acetylcholine will keep on working. When it keeps on working, that means this is a parasympathetic system. It is a parasympathetic system, acetylcholine. So acetylcholine will keep on working. Right? Now, when it keeps on working, how will you diagnose? When the parasympathetic stimulation is there, what would be the condition of pupil? What will be the condition of pupil? It would be dilated or it will be small. Think it like parasympathetic is rest and digest. आराम से खाना खा के will feel like sleeping और बड़ी-बड़ी आंखें करके देखें. We make sleeping, right? This means it would be a pinpoint pupil. This is the superb diagnosis. Pinpoint pupil. So easy to understand. Right? So whenever you think like that, someone has got this organophosphorus poisoning, first thing you just watch the pupil. The pupil would be pinpoint, small, shrunken pupil. Second, this is parasympathetic system. Parasympathetic system is rest and digest. So there will be increased secretion. Every secretion would be increased. Every secretion, you'll find that from mouth, the saliva is dribbling out. Because you know that in parasympathetic, it has to digest. So all secretions, they are raised. Parasympathetic is the system when it will say that yes, person can go to washroom, right? So that's why over here, there won't be any control on urine, right? So there would be secretion from there, right? HCl increase in stomach everywhere, right? The entire system, yes. So this is when there would be increase in all secretions. In all secretions. Not from the important from the perspective of exam, but because say from the medical point of view. Have you heard the name DDT? Before some time, right? They say that in the news that खेतों में DDT, they are spreading the DDT. Right? Okay, some have heard, yeah. Yes, Ms. Khan. Yes, fertilizer, exactly, fertilizer. Now this is DDT. It is diphenyl. It's chemical structure. And the trichloroethylene. DDT. But nowadays, you are not hearing of it. This was the first generation of very, very Purana. So that was, now see how things really change. This was organochlorine. Now, organochlorine is also one of the group of organophosphate insecticide. But then came the next generation. And they said, Kabhi, now with this, those insects, they are not dying because they have developed resistance. So there came the next generation. Next generation. And it is this next generation which is organophosphates. So all these thiones, right? When you hear malathion, so all these thiones, right? Phenitrathion, phenitrathion, these are all very powerful insecticides, pesticides. They use it. They use it extensively. And now, even with drones, they really spread everywhere. So these are the things which has got very bad effect on the neural system. Very bad on the neural system. Some say, I know few students, when they said कि सर हम कुछ भी करें पूरा दिन नींद ही आती है every day, right? It's like they are like there is no energy. It is like if the parasympathetic system is constantly triggered, right, so then person won't be able to pay attention. He'll feel like energyless. All these are organophosphorus. Right? These are organophosphorus. Poison means when the dose is so high that it starts giving major effects. But even in the low doses, right? Next gen is next generation. Next generation of these pesticides. After that, they have launched even more. They are the carbamates and organochlorines and chlorophenols, all extremely powerful. Extremely powerful because they said that they need to increase the production. But so dangerous. Yeah. So this is organophosphorus is one poisoning, and now comes the second poison. This is the last topic for it. And this thing is the world's most poisonous thing. Right? This I'm not comparing with potassium cyanide. Right? Not with that. The natural form of toxins. Right? This is the toxin. And this is called as the botulinum. When I'll tell you the lethal dose, you'll be surprised that so less. Right? Let's start with that one. That botulinum, right? I'll explain you. But you know how much poison is needed to kill a person if it is to be given orally? Right? Oral, the lethal dose that is sufficient to kill even a very powerful person. It is 30, not milligram. Not milligram, nanogram. Nanogram. It is that less. Inhalation. If person smells it, how much can really? Just 0.9 microgram. This much is sufficient, and it has got no taste, no smell, nothing. It's that dangerous. This is the poison which is generated by those anaerobic bacteria, right? Anaerobic bacteria. Anaerobic means they don't require oxygen. They develop in the absence of oxygen. Aerobic means oxygen is needed. Anaerobic means oxygen not needed. In that situation, they proliferate, they multiply. So it means that, and where, by what? It is this is the name, Clostridium. When we'll be learning bacteriology, all these things will come, right, in full detail. But just to know, Clostridium botulinum. So this is the bacteria. Now, up till this point, oh yes. Unfortunately, yes. If they come into contact with this, the death would be so bad. And as you have asked this question, so I'll talk about the treatment also, but it is heartbreaking if it comes, if anyone comes in contact. I'll explain that what happens. So first, let's know that where it connects or what it really does. This Clostridium, it acts on snare protein. It acts on snare protein, that is synaptobrevin syntaxin. What if those proteins, they are broken, gone? Now see, everything is fine. The vesicles, right, they have got acetylcholine. Receptors are ready, now, right, acetylcholine and we contract. But those who are taking it near the heart, so there is no exocytosis, right? Exactly. There won't be any no exocytosis. Everything was there, but it will not be released. And this leads to what is called as the paralysis and the flaccid paralysis. The person, the moment they come into contact with this, right, they become absolutely paralyzed, no movement, and then the respiration stops. Exactly. The mustard gas, sarin, all these nerve gases, they also work on the same principle. You're right, Nisha. But in mustard gas or sarin, you need slightly bigger dose. This is in microgram and nanogram. Is it a painful death? Person won't even know, right? But yes, it is like in spite of air, person won't be able to. So that way we can see that a painful death. So here, synaptobrevin and syntaxin, so no exocytosis, so acetylcholine is not released, so that will lead to flaccid paralysis. All the muscles, they will become bilkul weak, right? It is the flaccid paralysis. Now, where do we see this thing? Where do we find this thing? Being a doctor, you should really, after how much time the person will die after? So minutes. So now, Neha, you will get the answer. Where do we see these? Where do we find this? This is present, this particular bacteria, because see, it is like anaerobic, na? Anaerobic. So where there is no oxygen. So if there foods at risk, which are the foods at risk? One, canned juices. Canned juices. So that's why, say in US, etc., so they have got such a strict control, but not everywhere. So many times canned juices or those canned sauce, right? Sauces, they are. Then this is, say, the food wrapped in metal foil. Say if it is cup noodles, like that. Say over here, it's a food wrapped in foil. If it is immediate, so nothing would happen. Right? If it is freshly made and wrapped in. But if someone is consuming, right, sometimes people go for picnic or hiking, and then it is kept over there, and then after some time, when it is consumed, who knows that the bacteria might have started into that. So these are the things which are at high risk. Our Indian food, sometimes they ask the question that, say, our Indian food, in Indian foods you'll find these things less. That is a home cooked food. Right? In home cooked food, you will not find these things, because the best part is that if you boil, right, these are the two very good things: boiling and the pressure cooking. Pressure cooking, it kills Clostridium botulinum. It kills Clostridium botulinum. But remember, not the freeze thing. That if someone has got a fruit and it is kept in the refrigerator, then that is no guarantee. Freezing this bacteria will lavishly proliferate. Right? It is only the boiling and the pressure. They can only, in mess we get roti spoiled, but they must be fresh. Made on that day only. That will not be a problem. But so that is fine. This is when bacteria should get some time, at least 24 hours. So that's why I said that at times people take that when they go on mountains, right? So they get kitna sara food wrapped in the foils and it is over there. So though the temperature is less, but freezing has got no effect on this. So just freezing does not kill. Clostridium botulinum. It is only the boiling and the pressure. So that kills it completely. Now, what if someone has suffered? You find that Clostridium botulinum and you are into hospital and someone has consumed and patient is right in front of you, you got the antitoxin also. So treatment is antitoxin of this Clostridium antitoxin. Unfortunate part is that when this is done, even when the antitoxin is done, whatever the damage which has happened, right, whatever the damage which has happened, it is not reversed. Right? So this antitoxin, even when it is given, it does not. The only thing is it will prevent further damage. Prevent further damage. That's the only thing for which this antitoxin is. No, glaciers melting can have this poison. No, glaciers, so they don't have any food. Glaciers, वगैरह पे तो there is nothing. Glaciers, so they are just ice. Boiling also kills bacteria or germs present in. Same in the milk. That's true. So any of these things which are like the bacteria, and this is typically anaerobic bacteria. So which part is damaged? Well, over here, it will lead to flaccid paralysis of the entire body. And when there is flaccid paralysis, again, the respiratory muscles will be affected. The person will die. That is, in spite of the presence of air, person will be able to breathe. Because when you breathe, right, so this is an active process. Exhalation is a passive process. You don't have to do anything. Muscles, they are trying to relax, right? Lungs, they are trying to collapse. So air automatically comes out. So the principle of Python. See, I told you that, say, if someone is, the question which is asked is for milk. So when people make milkshakes, so they don't boil the milk. They simply use that Amul Taza Tetra Pak milk. So what about it? One is that that's why there is a very strict expiry date. Very strict expiry date. And whenever these Tetra Paks they are made, those Tetra Paks, because I've been to one of the factory and they said that there is a surface or an extremely high temperature and milk is passed through that surface at a very high temperature and then it is immediately packed. So when it passes through that surface, so nothing, no bacteria, nothing releases over there. The problem comes when you open the Tetra Pak and then after opening it, if you're not consuming it within 24 hours, then the trouble starts. Because anything which is freezed, it has got no these bacteria, so they can proliferate. So this much was for today. As I said, we'll go for the high-speed revision. So remember that. We revise within 5 minutes. All right. Here is the neuromuscular junction. This is Sharrington. Mr. Sharrington is considered as the father of synaptic transmission and the father of neurophysiology. So these synapses, they are the modified synapses. Sympathetic, parasympathetic, we talked about spinal nerves, total 31 pairs and 8, 12, 5, 5, 1 respectively for the cervical, thoracic, lumbar, sacral and the coccygeal. These nerves which will be coming out, right, coming out in between the vertebrae from the spaces, and as they come out, they will be, if we take the cross section, that cross section is, point to remember that it is the posterior part, that is afferent, and it is the anterior part, it is the efferent. Efferent is motor, which will go towards the muscle. Afferent is sensory. Finally, there is a common path, right, in which all the nerves, they come out after coming. When the spinal nerve comes out, then there is anterior and posterior division, right? But we'll see that later. But over here, it is a common pipeline through which the afferent and the efferent, they are entering into this system. So here it is. There is sympathetic ganglion. This sympathetic ganglion, which will be playing a role into the autonomic nervous system. So here it is, the cervical and the sacral, they are parasympathetic, and thoracic and lumbar, they are sympathetic. The action potential, there is sodium potassium pump and the potassium pump, it is ATPase. So they are maintaining this resting membrane potential. As the impulse comes, there is sodium influx. It starts rising. When it reaches to -60, there is trigger of voltage-gated sodium channels. Then at its peak, sodium is inactivated, voltage-gated potassium channels open, they drop all the way, and there is hyperpolarization. And sodium, potassium, ATPase pump and potassium, they reset the whole system, and once again ready for the next. The myelin sheath, and then the action potential, which is generated over here, it continues all the way up to the end terminal. At the end terminal, the action potential comes over here. We have got the vesicles. These vesicles, they are filled with the acetylcholine, which is a neurotransmitter. We'll be requiring two things. One, the calcium. Second is the snare proteins, which are the surface proteins on the vesicles and onto the cell membrane, so that they both will be coming near each other and will be leading to exocytosis, so that this acetylcholine is released into the synapse. This whole process is called as exocytosis. These are the two things which are needed. Snare protein, synaptobreviin, which is on the vesicles, and the syntaxin, which is on the cell membrane. They learned two important principles over here, that all the cell proteins whatsoever are needed, all, right, they are made into the cell body, and then they travel all the way to the end, and this is where they will be working. It is only the acetylcholine which is formed at the terminal, right? Rest all things whatsoever is needed comes from the top factory. Our mission starts from GIT. In GIT, whatever we consumed after metabolism, this choline is formed, it comes into the circulation. From there, it comes into the ECF, entire body. And then we have got our first protein coming up, it's choline transporter. This choline transporter is a surface protein, and it takes the choline in. Mitochondria is there. Its job is to make acetyl coenzyme A. They both combine in presence of choline acetyl transferase, and then there is formation of acetylcholine. This acetylcholine is immediately deposited into the vesicles, right? That is by transporter protein. Once again, this transport protein, that also comes from the top only. So they are now ready. Now the guns are loaded. On to the next. As the depolarization wave comes from the top, there is depolarization sensitive calcium channel. So basically, voltage-gated calcium channels, right? Technically speaking, voltage-gated. But we say depolarization sensitive calcium channels, so that clarity is clear that when the dipole wave comes, they open. It activates this calcium sensitive protein, and this calcium sensitive protein will start telling that go towards the cell membrane, go towards the surface. So they all start towards there. That's where the synaptogravin and syntaxin, they hold hands and they start pulling. And as they start pulling, right, finally the layers are merged and then it opens and the acetylcholine is released. But not to waste anything, so clachrines, so with clachrines, these are once again proteins. They lead to recovery of these vesicle walls, right? And the vesicle is formed again. Whatever the deficit is there, that is filled by the new production. Acetylcholine, right, should not keep on acting like we saw in some of the cases of poison. So in acetylcholine, it is broken down by cholinesterase, right? Cholinesterase to acetyl and choline. Acetyl is easily available, right? Mitochondria will be making. So it is diffused out, but this choline is recycled, right? So it is reabsorbed back. And then nicotinic receptors, they are stimulated because for them this would be a ligand. So this is ligand and it generates the action potential. Clinically, talking about the pufferfish, which leads to this tetrodotoxin, this is fatal because it blocks that voltage-gated sodium channel. Voltage-gated sodium channels, no action potential, no release of calcium, no acetylcholine, no muscular contraction, and respiratory muscle paralysis and death. This is the outcome in all the cases, right? Because we are dealing with neuromuscular junction. So in neuromuscular junction, it is blocked at any of the level. This whole cycle, any place it is broken and the patient gets that trouble. This is where we talked about something like our immune system, that is the antigen. Antigen is those pathogens who are generating the pathology. They are generating something which is abnormal, bacteria, virus, fungi, parasites, like all. So immunoglobulins, that is a fancy technical name of the antibodies, right? So IgG, IgA, IgM, IgD, IgE. But this takes about 7 to 10 days. When it takes 7 to 10 days, that means it will take some time. That's why this immunity is also called as the adaptive immunity, right? Adaptive immunity. Adaptive immunity means it takes some time before it really acts. The first line of defense, when we have used the word adaptive, the first line of defense is what is called as the innate immunity. I-N-N-A-T-E. Innate immunity. It's the first line of defense. So the moment it is seen that there is some infection, there is inflammation, immediately this innate immunity will come into picture. And in the T cells, T cells, B cells, and then there are natural killer cells, lymphocytes. So those natural killer cells, they are the first line of defense, they come into picture. But over here, say we took the example of pluripotent cells, those cells which will be generating, they are present into the bone marrow. They have got the PIGA gene. That PIGA gene which will tell these that all bachas, remember, when the complement comes, cut it, and here is the knife for it. This is what is called as the surface cutter or the complement destroyer protein, which is gifted to all of them. So every RBC, every WBC, every platelet will be having that. So the moment complement comes to eat it, they cut it, right? But these complements, these PIGA genes, they are not present into the bacteria, virus, parasites, fungi. So that's why these complements, when they will be going to bacteria, so there they will be the boss, right? So it is like that these are the gundas, right? These are the gundas which will be acting against someone and they won't be able to act against us, right? So that's the complement system, because if the RBC is broken, then it leads to hemolysis, hemoglobin is released, taken up by kidneys, from kidneys it will go into the nephron, in nephron it will become proximal convoluted tubule. Once it comes over here, proximal convoluted tubule, bichara innocently will feel like, okay, this is a protein, let me take it back. As it takes it back in the interstitial space, it is broken into heme and globin. This iron is super reactive, poisonous for the tissues, breaks the tissues, destroys kidneys, and it is what is called as the tubular necrosis. And then obviously it is not only the hemoglobin, technically speaking, even the WBC, platelets, all those things they will be going. And when you go for the urine test, you will find that in such patients there'll be blood in the urine, hematuria, there'll be hemoglobin in the urine, right? There'll be hemoglobin into the urine. So all those things, they are the evidence of tubular necrosis. So in case of autoimmune disorders, they make antibodies, but these are the antibodies against the own cells, against the own cells. That's why we call them autoantibodies. So there is one condition called Lambert-Eaton syndrome, where it will be affecting the voltage-gated calcium channels. So this is a presynaptic disorder. Voltage-gated calcium channels, when they are broken, done, finished, right? Acetylcholine will never ever be able to be seen because their calcium sensitive proteins will not be activated. So there won't be any activation of synaptogravin or syntaxin. So they will never go on the surface. Other case, myasthenia gravis in age, as the age increases, you must have seen many of them. There was some Amitabh Bachchan's movie also. He was suffering from myasthenia gravis. So gradually the movements become very slow, right? Their hands, they keep on shaking like this. Handwriting starts becoming very bad, right? Because handwriting is one of the very sensitive thing to figure out the myasthenia gravis. But in medicine, when we'll be talking about it, that there are some very early signs. One of the early signs is this the handwriting, right? When the handwriting starts coming bad. So there is formation of autoantibodies against the nicotinic receptors. And this is a post-synaptic disorder, muscle weakness and paralysis. Then we talk about those poisons. One of the poisons is our phosphorus poison, pesticides. And these pesticides, they block cholinesterase. So once the cholinesterase is blocked to acetylcholine, it keeps on acting, right? There is overstimulation of parasympathetic system. So pupils are pinpoint pupil, and there is increase in all secretions. DDT, this was just dichlorodiphenoyl ethylene. This is the organochlorine. This was also dangerous, but it was less. The newer one, this malathion and phenyltoluene, they are the very powerful organophosphorus. Pesticides, very powerful pesticides, and they are considered as the next generation of the organophosphorus. Then finally, the botulinum. This is released by anaerobic bacteria. Anaerobic means those who proliferate without the presence of oxygen. So in such cases, they proliferate when there is no oxygen. Lethal dose, oral just 30 nanogram, inhalation just 0.9 microgram. Intravenously, it is 0.08 microgram if it is intravenous. It is that less. Right? There is no smell, no taste, nothing. Right? It is by Clostridium botulinum, and this is where it is breaking synaptogravin syntaxin. So there is no possibility of any exocytosis. Acetylcholine is not formed. It's to flaccid paralysis. Foods which are canned juice, canned sauces, food wrapped in foil. Freezing would not kill this bacteria, but yes, boiling and the pressure, that surely kills this Clostridium botulinum. Right? When if the patient is lucky and if it's available and you give antitoxin, botulinum antitoxin, but whatever the damage which has occurred, it is done. Right? It cannot be reversed because it leads to permanent damage, but further damage it can be prevented. That's why these things, they are pretty dangerous. All right. So that's it for today. Thank you so much. Have a very nice weekend. Enjoy, study, and thank you. How can I join? USM. अभी तो I'll let you. Let the time come. I'll definitely. Don't worry. Okay. See you and meet you soon.